So, good morning and warm welcome to everyone here. So, we are honored to have Dr. Sonia with us as our distinguished speaker today. So, this webinar is a part of the series that is known as the Interdisciplinary Green Equation series. This series is a knowledge sharing initiative aimed at fostering collaboration across diverse scientific domains that addresses challenges in sustainability, innovation, and environmental stewardship. This series brings together experts, researchers, and students from life sciences, environmental studies, agriculture, biotechnology, and allied disciplines to explore green solutions for a sustainable future. Through focused webinars, lectures, and interactive sessions, the series emphasizes integrative approaches, blending science, technology, and innovation to advance research and practical applications for ecological well being and resource efficiency. I am honored to introduce Dr. Sonia Malik, ma'am, who is a professor in biotechnology and Dean of Research and Development at Baba Farid Group of Institutions, Bathinda, India. She is specialized in plant tissue culture, genetic transformation, and production of specialized metabolites to be used in pharmaceutical, food, and cosmetic industries. Dr. Malik held various academic and research positions at Brazil, Czech Republic, Australia, and France. Her research work is focused on enhanced and sustainable production of bioactive compounds from plants using different biotechnological approaches. She has more than 50 research papers and is the editorial board member of many international scientific journals. Welcome, Sonia, ma'am.
Thank you, Dr. Panna, for your nice introduction. So now let me uh, share my screen. Is my screen is visible? My screen is visible?
Not
Is
yet,
okay?
ma'am. No,
Not
ma'am.
yet?
Not yet.
Ma'am, first click on upload arrow sign, share screen, then select the option entire screen, then share.
Now?
Yes,
Okay. Is okay?
it's okay.
Okay, fine. So today I'll be talking on from cells to plants, plant tissue culture in specialized metabolites production. So in this area, actually, it's a quite vast area, and here I have divided my talk into 5 subparts. First, about the brief, not very brief. I mean, I wanted to give you the background of what exactly plant tissue culture is, and then before knowing about the plant tissue culture, what are the plant natural compounds and why do we need them, why plants produce them, all we'll discuss about this. And then what are the requirements to start the plant tissue culture, what are the benefits of producing these compounds using tissue culture, and then I have tried to give some case studies and the work I have done during my past, as my past experiences or job, and then finally the conclusion. So, as all of us know, mostly all of us know these days what are the natural compounds. And if we think of the three main challenges of today's world, these are climate change, health, and sustainability. Global warming and climate change is one of the main issue these days, and then health. Everybody is looking for natural compounds because these are better, they have the better compatibility with the human body, and we are using them since very long time ago, and there are many side effects with the synthetic drugs or chemicals. So now people are coming, I mean, they, with the resurgence of public interest towards these natural compounds, the demands for these compounds are also increasing day by day. If we think of even cosmetic, if we talk of agriculture field and pharmaceutical, everywhere, everyone is looking for the natural compounds. Now, another challenge we are facing in today's scenario is the sustainability. I mean, how to develop these compounds without in future, from where to procure them for their constant production. If we take them these compounds from the plants, it will lead to loss in biodiversity. So these are the main three global challenges, and even these challenges they have been covered by the sustainable development goals also, as raised by the United Nations, and all the countries are involved in it, and this is the worldwide agenda to fulfill Sustainable Development Goals till 2030. Sorry, I would like to say here, if someone wants to ask something, he or she may unmute self or herself and ask if something is not clear. And now various applications of natural products. Now let's think of the plant. We are dependent on plants since from our birth till death. When child is born, he needs the cradle that is made up of wooden, and that is made from the plant wood. And then for the food throughout our life, rice, grain, vegetables, fruits, we are dependent on plant totally. For till death, at the end, the tomb we need, that is also made up of the plant. So here we say that we are dependent totally on the plant since time immortal. Even our ancestors, our forefathers, our grandparents, great grandparents, they all used to heal themselves with the drugs, or the not the drugs, we should not use this term, the pharmaceuticals or the traditional medicines obtained from the plants only. And if we remember, like long time ago, when the little child, the small child, like the newborn, or within 6 months or 1 year, he or she get cold, cough, only home remedies they were used to be, they were treated with the home remedies like made from the fennel seed, from the plants bark or the flowers, or any, even from the asafoetida. These are all the plant species commonly used, curcuma, all the compounds, they are obtained from the plants. And in still in many countries, especially in the developing countries, people, more than the 80% of the population is dependent on the plant-based products, on the natural medicines to cure themselves. So in developed countries also, now the people, they are developing interest to use natural or plant-based compounds because of their lesser side effects and better compatibility with the body. And now here, if we look at the area of pharmaceuticals, they are used in the life-saving medicine like Taxol and Chiconin, that we are going to discuss in our further slides, and food items, and even the ginger and all, like during the cough, cold, our mothers give. And then in case of after food nutrition, also in agrochemicals, in the area of agriculture also, these natural compounds derived from the plants, they protect crop in the form of natural insecticides, and they are proved to be better than the synthetic insecticides or pesticides, because no doubt they can help in enhancing the yield for a time being, for the moment, the synthetic pesticides. But after all, their side effects are too many that lead to many diseases like cancer, asthma, or all they are due to the use of this synthetic. And now in the area of cosmetics also, there is more interest day by day to use the cosmetics with antioxidant properties, antimicrobial properties within the creams, lotions, to protect the natural skin biota, or to improve the hair or the texture of the skin, etc., and even for the to reduce the cancer disease. So now I think with this background, if we look at the market value or the total annual turnover with this natural compound, the food items, it accounts for 85 US billion dollar, followed by cosmetics, and then pharmaceuticals, agrochemicals, and pigments like used in the food, as well as to dye the cloth or any other items, they account for the 8.53 billion. So now by looking at these figures, you can get an idea how important these are, and their value is going to be increased by 12% more in next coming 10 to 15 years. So this is the slide, the picture showing the natural compounds, and they're used in various areas, as already discussed in the last slide. So here we can assume, or we can imagine the value of these natural compounds in the global market. And now if we go a little deeper into this natural compounds market, we may say that 25% of the natural drugs or the pharmaceutical companies, they are 25% of the drugs, they are still formed from the plants. Either they have dependent wholly on the plants, totally on the plants, or part of them or the compound in the chemical synthesis. They use just the part of the compound, or the basic or the initial structure or the initial components is derived from the plants. And there are 121 active compounds. They are in very much use in medicines related to cancer, malaria, or other life-saving drugs. And here, if we expand the market of pharmaceuticals, the annual market or the global market, sorry, or annual rate of the global market, it accounts for 114 billion by 2030, which is, as the figures shown in the last slide, it is going to raised by too much amount with the cancer treatment from the drug Taxol, which is the main drug obtained from the Taxus brevifolia. This is the single plant species which is used for the cancer treatment, and we'll see this in the next slides. In the case studies, I am going to show you how many plants are required or how this drug is obtained and what are the different pathways to obtain this Taxol from the plant species. And then the malaria treatment drug artemisinin, that is also derived from artemisia plants. And now coming to the food industry. In food side also, traditionally we are using fruits, vegetables from the food, but the modern food industry is making the steviol glycosides. Those are the low calorie sweeteners from the stevia rebudiana plant species to control the diabetes. I mean, in the diabetic patient can not to control is not the right term. The to in case of diabetes, the low calorie sugar pills, they can be obtained. They are used, and they are obtained from the stevia plant species. Similarly, antioxidants and other polyphenols from the apples or coffee, they are obtained from the different plant species. And the modern food industry is dependent again on the bioactive compounds from the plants in from the different species and related to the different families. And now in agriculture also, like the you have might have heard the name of the neem, very common tree, as a directin, that is also used in the pesticide to control the number of pest diseases. Even at home also we use them to preserve our clothes, the winter clothes. And there are other antifungal saponins to control the diseases, fungal or bacterial, in case of strawberries. And now this is the overall scenario in case of cosmetics. There are many compounds, natural compounds, for instance, chiconin, resveratrol obtained from the grape, and chiconin from the Lithosperma species or the Arnibia species, and arbutin, and there are many more. These are the just common examples which are the commonly used. Now I am interchangeably using the different terms. You might have heard sometimes I'm using the specialized metabolites, bioactive compounds, or secondary metabolites. So now let me make you clear about all these terms. What exactly are the plant specialized metabolites? These are actually nothing. These are the plant natural compounds only. Just to make them more defined term, they can be used interchangeably. All the things derived from the natural sources are called the natural compound. If the source is plant, then we call them the plant natural metabolites. If the source is from the animal, we may call them as the animal derived natural metabolite. And to be more specific, we use the term plant secondary metabolites also. But recently, the term secondary has been replaced with the term specialized metabolites, because as the plant produce two different kinds of compounds, primary metabolites and secondary metabolites. As the name suggests, primary metabolites, they are obtained as a result of the primary metabolism occurs within the plant species. For example, the plant make food with the help of sunlight, chlorophyll, and water, and as a result sugar and other carbohydrates or proteins or whichever things they are produced in the plant to survive themselves, to survive them, they are known as the primary metabolites. And apart from those primary metabolites, plants also produce secondary metabolites, or we may call them specialized metabolite or bioactive compound. Those are actually not required or essential for the plant's growth and development, but actually plants cannot survive without them. They are required for the fitness of the plant species to combat them with the natural calamities or number of diseases. As they are sessile, they cannot move from one place to another place. For example, drought changes or another kind of natural or environmental changes, they have to protect themselves by producing these compounds. These compounds may sometimes act as a signal also to ward them off from some of the diseases also. So plants produce because of them to protect themselves from the environment. But these specialized metabolites are useful for us also, as depicted in the last slides. We use them as a food, cosmetics, insecticide, pharmaceuticals, different medicines, natural dyes and fodders. Now, if we look at the estimated retail price values of these different specialized metabolites or bioactive compounds obtained from the plant, and here if we look at the vinblastine and vincristine, these two compounds, they are obtained from the periwinkle plant which is a herb species grown commonly alongside our houses. I'll be showing you the picture of this plant in the next slides, and they are $2 million. And similarly, vinblastine also, these two are used in the cancer treatment. And similarly, Taxol also zero point 0.6 million US dollar. And then capsaicin obtained from the capsicum, the shimla mirch that we eat, and digitoxin and colchicine. These all are the different compounds used in the different areas in the food, cosmetic, or most of them they are commonly used in the medicines. Now, what are the different challenges and risks to obtain these compounds from the plants? Here, one thing is clear, that these compounds we have to get from the plants. So, to get these compounds, naturally we have to extract the plants parts. Like for example, if some compound is present in the leaf, then we need to obtain the leaf from the plants, from the stem, root, buds, flower, whatever part it is. Ultimately, we need many kilograms of plant to obtain just a few percentage of these components, because as I mentioned, plants produce them just as a kind of a signal to protect themselves from the environmental diseases or the environmental calamities, and they are produced in very low quantities in the compounds because they are very poisonous and toxic, and plants produce in very low amount, that too in a very specialized cells or organelles in the membrane bound organelle. And as they are produced in like 0.01% dry weight, for example, Taxol yield, that too after 5 to 6 years. So you can imagine how many number of plants we have to sacrifice just to obtain 0.01% this Taxol. And as the demand is rising day by day, so now the question arises how to obtain this sustainable production of these compounds, that too with the increased or the enhanced amount. And now again, big challenge is the seasonal variability. All of us, we are in different globally, we are facing this climate change challenge. So if the particular compound is producing in, let's say, at the cold temperature or at the extreme hot temperature, if the global climate is changing globally, then how can we imagine the production of these components in a very homogeneous manner, just within a in sustainable amount and in high yield? And now there are many other environmental factor also. For example, if particular compound is producing at, let's say, at the temperature of 35 degrees Celsius, and the another coming years, if temperature rises to 1% or I mean the or the 2 degrees Celsius, then it is very difficult to imagine the production of these compounds. And just a moment, just a moment. Yeah, sorry for the yeah, sorry for the intervention. Yeah. So now another biggest challenge is the overexploit. As to obtain very little amount, we have to sacrifice large number of plant species. Now, where the biodiversity will go? On one side, we are looking to conserve or save our biodiversity resources and how to conserve them. And by taking out all these plant species and sacrificing them just to obtain a small amount of drug or for our use, where the biodiversity will go? So how can we fulfill these sustainable development goals? So there are number of risks. So with this in mind, now I give you another example to make you more clear, like what I am talking, it will be more clear with these coming slides. Let's take the different examples from medicine side, from food side, or from pigments. Take the example of Taxol. It is very well known anti-cancer drug, and it is obtained from the yew tree, and the botanical name is Taxus brevifolia. And it is this drug, this is approved by Food and Drug Administration, FDA, and it is obtained from the plants of this yew tree species. Point it 0.1% to 0.01% in the dry bark, the bark of this species, that too after 6 to 7 years. So that means we have to grow the plants and wait for 6 to 7 years, take out just only the 0.1% to 0.01% of the this taxol. And once we take out the bark, the plant is going to die. And for obtaining 1000 kilogram dry bark, just to obtain 1 kilogram of taxol, we need to have approximately 8 to 9 trees. Just to obtain 1 kilogram of taxol, we need 1000 kilogram dry bark of this plant species, and to obtain 200 milligram of taxol, 8 to 9 trees are required. And 200 milligram we need just to treat only one single cancer patient for the 6 to 7 cycles, chemotherapy cycle. And now you can imagine where the biodiversity goes and how can we obtain the number of these plant species. And you can also relate these figures with the cancer patients worldwide we have. And now the annual turnover of taxol is 1.6 billion US dollar. Now another example, this was the example of from the medicine side. Now the another example of periwinkle plant that I was talking in my previous slide. This is the also known as the Madagascar plant, and these flowers are commonly you can see growing along the roadside or beside your houses or and this plant species produce vinblastine, vincristin, vindoline, agemalacin, different components in flower, in leaves as well as in the stem. These are commonly known as the vinca alkaloids because this belongs to the category of vinca alkaloids. Here I didn't show you the details of the secondary metabolite. We can divide them or classify them based on either on their uses, like whether they are going to be used in the food or in agriculture or medicines, or we may also classify them based on their biosynthetic pathways or based on their chemical structure. And based on their chemical structure, these plant species, they contain the alkaloids which are commonly known as the vinca alkaloids, and they are also the cancer fighters. And medicines, some of the medicines, they have the components from these bioactive compounds from this plant species to treat high blood pressure and diabetes, and they are also used as disinfectants. And we need 530 kilogram of fresh leaves to obtain 1 kilogram of vinblastine and 1 gram of vincristin, the pure form of these compounds. For these we need to have 530 kilogram of fresh leaves. That was the example of another example of medicine side. And now coming to the cosmetic side and the pigment side and the food side. This is the one of the example of the chiconin, which is commonly used in the cosmetic also, and it is also used as a flavor or fragrance in the food as well as coloring clothes also. So this is the once in 1985 there was a big hit in the cosmetic market with the bio lipstick. This lipstick is made up of chiconin, and in spite of its high price $30, because it contained the antibacterial properties also, and it obtained from the plant species that are confined to Korea, Japan and China. And to obtain this chiconin, the plant material is very less. That's why to make this lipstick, the cost incurred was very high. So that's why the single stick of the lipstick cost around $30. And in spite of its high price, 2 million pieces of sticks, they were sold within a few days by Canibo Japanese cosmetics company. The idea behind this lipstick was that it is not only biologically prepared, but also it acts against bacteria and has many other antimicrobial activities. It is also used in food pigments, and the same as in case of Taxol, it reaches a maximum production of 1 to 2% in the roots, that too after 5 to 7 years of growth. And to obtain this 1 to 2% after 5 to 7 years, we need to uproot these plants because this component is present in the root. And once we uproot the plant and take out its root, plant will be no more survive. It will be dead. And these are some of the components present or the compounds or the product available in the market using these components: ointment, lip balm, soap, burn cream, moisturizing lotion. And this is a Chinese company, Spring Wind. It is using this component from the plant. And this chiconin is obtained from Arnibia euchroma, but originally it is derived from Lithospermum erythrorhizome, And this plant species is confined to China, Japan, and Korea. And this plant is almost extinct due to uninhibited collection from the wild, and even the cultivation of these plants, they fail to provide the sufficient raw material. And due to some geographical regions, this plant species cannot be grown in the another plant, the another areas. And annual consumption of this chicon is 150 kilogram. Almost 10,000 kilogram of lithospermum they are used annually only in Japan to meet the market demand annually. But still, this they are not able to fulfill the market demand. They are only able to produce the 75 kilogram, that to using biotechnological production, that I'm going to show you in the next slide. And now the question arises, where can we look in for the viable alternative sources for the sustainable production of these compounds? And here comes the role of biotechnology. And plant tissue culture is one of the aspect of biotechnology that transform the cells into the mini factories to produce these specialized metabolites all over the year in a sustainable manner and independent of the climatic and geographical conditions, because we use this tissue culture technology under the controlled environmental conditions and on the controlled media. Now, coming to the basic concept of plant tissue culture, before knowing more about this technology, let's see the what are the basics of plant tissue culture, about its history and all. Plant tissue culture is based on the totipotency. That means all the plant cells, single cell of the plant, they have the capability to give rise to full-fledged plant or regenerate into the complete plant if grown in the artificial media and given the proper environmental controlled environmental conditions. So these are the basics of plant tissue culture. And now the idea of totipotency, it was given by Haberland in 1902, and he proposed that all the plant cells are totipotent, and he is also known as the father of plant tissue culture. He was the German botanist. And now further Stewart proved his experiments and he proved the concept of totipotency by growing the carrot cells. He made the discs from the carrots and grew it on the artificial media, and he was able to see that these carrot cells, they are able to regenerate into the full-fledged plant. Now again, just to summarize, what exactly is plant tissue culture? Plant tissue culture means the culture of plant cells, tissue or organs under the artificial media, on artificial media under controlled environmental conditions. And controlled environmental, I mean to say that temperature, humidity, light, everything is controlled, and we have to provide them the aseptic conditions. That means there will be no growth of microorganism, and we autoclave the media and autoclave all the things or sterile the things to make them aseptic to avoid the contamination or the growth of microorganisms. And then here comes one term, explant also. Explant is the any plant part or the tissue or the cell which we are going to use as the beginning or the starting material in the tissue culture, that is called as the explant. It may be the leaf, stem, root, or even the undifferentiated mass derived from these organs or tissues, that is also known as the callus. And then comes the regeneration. So basically in plant tissue culture, we change the cycle or the brain of the cells, because if we just take the example of the seeds, they have got the embryo and they are destined to produce the shoot and root. But in tissue culture, what we say, if we take any explant, and it will definitely give rise to, it has something already predetermined cells which give rise to the particular organ or or go to the another particular stage. But in tissue culture, we de-differentiate them, we change their mind makeup so that they form the callus, and then according to our purpose we make them to regenerate either in the root or in the shoot or whatever we want them to produce, or the particular kind of specialized metabolite. So here we can say clearly the cellular reprogramming, because all the cells, they have predestined program, but we make the plant cells or organs to reprogram. That is the capability of the tissue culture, and that is the beauty of the tissue culture and what tissue culture is exactly. And that this is whole dependent on the hormonal control, because all the plant cells, they have some kind of endogenous level of hormones. Hormones are also known as the plant growth regulators. If they are naturally synthesized, they are called the hormones, and if they are synthesized in the laboratory, those are known as the synthetic hormones or plant growth regulators. They may be auxin or cytokinin. Auxins are important for the root development and cytokinin for the shoot development. And if the level of cytokinins that are responsible for the shoot formation it rises, then the plant will give rise to the shoot. And if the auxin level is high, then root formation will occur. But if somehow we make the level neutral, that means the same level as the auxin or cytokinin, then instead of giving rise to root and shoot, plant will or the organ will form the callus. And now, what are the different key factors which are responsible for the tissue culture success? Number very first or the very important part is the explant source. Like which kind of explants we are going to select or choose. It depends a lot on the overall success of the tissue culture, because if our initial material that we are going to take to raise the tissue culture plant, or we are going to get obtain the any plant derived natural compounds, or just to multiply them using tissue culture method, if our initial source is contaminated or not good enough, then we are not going to get any results. So the very first requirement is to have the explant which are very healthy and in the young dividing stage. And no doubt there are some sterilizing agents or the methodologies. After obtaining the explants, we have to treat them with the bleach, with the antifungal compounds, antibiotic, and soap to remove all the surface contaminants. And then culture media composition. Like it depends on which type of hormones, which type of nutrition components like macronutrients, micronutrients is required by the plants, or if we are looking for the production of particular component to be produced by the plant, let's say the chiconin or taxol. So we have to first study the biosynthetic pathway of the plant, like what is the backbone of this component and how they are synthesized biosynthetically, and accordingly we need to include more macronutrient or micronutrient for the production of those components. And it's always not the case, because there are many factors involved: genetic, endogenous level of these components, hormones, and there are some kind of synergistic effects also. So it's not that easy as it looked like. So we need to perform number of experiments, number of standardizations to set the protocol for not even for a particular plant species, but also for particular plant organ or tissue also. And then next come the environmental conditions also: temperature, light, and humidity, they also play their role. And not only the light, but the quality of the light also, whether we are providing them with the red light, blue light, and for how much duration we are going to provide them. So this also affects a lot. And if we get the contamination, then it will, like if everything is going well, but at some stage after 5 or 6 months, if we get even the minor contamination, then it is going to lose our work for that we have done for the past, even for the years. So the main important thing is to keep the sterile environment free of the contamination, and then choose and then the selection of the explant. And various applications of plant tissue culture are especially in the area of agriculture to conserve the germplasm. In this we, if some germplasm that is going to, we have the plant species or any species which is going to extinct, or the rarely known plant species, we can culture them and preserve them in the in vitro gene banks using cryopreservation technique for a number of years. And we can preserve this at -196 degrees Celsius. And then whenever we want, we can take them out and use them. So in this way, it helps in the conservation of biodiversity. There are number of protocols. It's not like that immediately we take out and immediately put in -196 degree, it is going to die. There are number of procedures how to preserve them. There are different kinds of cryoprotectants and chemicals we need to use them. And then while revival also, we need to bring them to the room temperature first, and then increase the temperature slowly, not in a single go, and thawing process, and then bring to the live stage again. And with tissue culture, we can obtain the disease-free varieties that are sometimes not possible outside. If we take the case of getting the virus free plant, and if some virus is there, it is very difficult to get rid of virus, especially than the bacteria. And with the tissue culture technology, by using apical meristems, it is possible to produce the virus free plants. Even the some plant is infected from the virus, we can take the apical bud and grow in the tissue culture, and in that way we can obtain the virus-free plants. And it also helps in the quarantine purposes. For example, if we need to take some plants across the borders, it is tissue culture plants. They are we can take very easily, and there are no any kind of documentations are required, because since they are tissue culture derived, so it is understood that they will be free of any contamination or any. And another is the rapid clonal propagation. For the fast multiplication, tissue culture is one of the best strategy. And for example, if some plants through the normal traditional cultivation method, from single explant or from the single stem cutting or from the single seed, we are going to get only the single plant, and that too in a year we can, if we have a big land, we can get some number of plants. But by using tissue culture technology, just within a flask we can get number of plants, that too within a few months. Like from a single explant or very small segment of the leaf, say 1 centimeter to 2 centimeter, we can obtain the one plant. And if there is some rare or endangered plant species and we cannot afford to take the plant material, we just need only one single leaf. On from that we can have a number of billions of plants within a year. That is the main application of plant tissue culture. And in case of orchid, especially orchid plants and other trees and the monocot, this tissue culture technology is very useful. But in case of monocot, those are recalcitrant to be grown in the plant tissue culture media, and research is still going on in that area. But in herbaceous plant, tissue culture method is very successful, and in tree species also, that is little hard than the herbaceous plant species, but it is not impossible. And then overcoming dormancy also, plant tissue culture technique is very helpful. For example, if some seeds they are not able to give rise to the plant, then we can get the embryo, isolate the embryo and grow them. And even it is possible to obtain the haploid plants using anther culture or microspore culture. And another one of the biggest application of tissue culture technology is to obtain the new variety. Through breeding methods, it take about 5 to 6 year minimum to obtain the new variety, but by using tissue culture, we can get the new variety within 2 to 3 years at the most. And now, again, just to summarize, we may say that plant tissue culture is a sophisticated technique that enables scientists to grow plant cells or organs in sterile and controlled environmental or laboratory conditions. And it uses the concept of totipotency. Whole tissue culture method is based on the tissue culture technology. Sorry, the totipotency. And now, as we now understood about what exactly plant tissue culture is, now coming to the media and what are the different requirements, nutrient requirements which plant need. Because if we look outside, plant is growing in the soil and it is getting everything from the soil through its root, whether it is nitrogen, phosphate, sulfur and other micronutrients, and in the form of fertilizers also we are providing them. But how to give these nutrients in the tissue culture? Because plant needs something, like we need food to grow, same way plants also need something to eat and grow. And then the plant, talking about the plant media, this I have already covered about the different explants. Plant media, it contain different elements. It contain macro elements which contain phosphorus, potassium, calcium, sulfur, etc., and micronutrients like zinc, cobalt, copper, and then organic supplements, glutamine, amino acids. These are not very essential, but these two components are very essential part of the nutrients. And then different kind of media are devised based on the nutrient requirement of the different group of plant species. Most commonly used plant media is the Murashid and Skoog media. It is generally commonly used plant media, and for the tree species, woody plant media has been devised, and other copper and nick media is also devised for the hydroponic cultivation. And now either there are two methods. We may prepare the stock of these nutrients, because there are number of components, and it's not like we are going to use the phosphorus, potassium directly. There are the salts like potassium nitrate, sodium hydroxide, calcium nitrate, and we are going to use the salts. And there are the different micro level components. They are already devised in this different media, and the concentration we have to use accordingly. There are the number of catalogs which are showing the details about this media, and either we may prepare the stock and keep in refrigerator to use for 1 to 2 months. But now these days, ready to use media, they are also coming, and they are very convenient and handy. We need to just take the in the powder form just to weigh. It's mentioned on the packet or the bottle how much you need to add in the 1 liter, and even the no need to set the pH also. It's everything is set. We just need to dissolve it in the water and autoclave it, and it is ready to use. And in the earlier time, or if someone cannot afford that media, ready to use media, he may use the stock and keep in the refrigerator for the. And now another important point in the media is the important thing in the media is the carbohydrate source. And it is in the form of generally in the form of sucrose, because the plant they need mainly they're all the metabolism, they're dependent on the carbohydrate source, and in the outside they make their own food and they don't need this sucrose and all, because they are utilizing the chlorophyll and the water and sunlight. But sunlight we are providing, but the plants they are not matured enough, and they don't have the capability to utilize the already available. I mean, their system is not that much developed. So we are providing them each and everything, and also we are manipulating them according to our interest. So main important thing is the inclusion of the carbohydrates, and carbohydrate may be either in the presence in the form of sucrose, glucose, galactose, fructose, but commonly used carbohydrate source is the sucrose. And after autoclaving sucrose, it breaks into fructose and glucose. And generally, plants they are capable of utilizing the carbohydrate in the form of the glucose. And if we add the sucrose and after autoclaving, it break down into the different components, glucose and fructose, that easily utilizable by the plant. And in the tissue culture media, we generally use 2 to 3% of sucrose. And now coming to the pH, that is the very critical medium parameter. Generally, pH need to be keep at 5.6 to 5.8, because pH it controls the nutrient solubility also. All the nutrients which are present in the media, if we are not providing the particular pH 5.6 to 5.8, the nutrients will not be able to dissolve. And another important thing is the plants they will not be able to absorb the nutrients also if the pH is not right. And it also give the proper solubility, as well as it helps in solidification of the media also, because the tissue culture media needs to be kind of a semi gel like media. And to provide the support, we use different gelling agents, and agar is the most commonly used gelling agent. It is derived from the seaweeds, another plant, red color algae which is growing along the seaside. The agar is obtained from that. And Gelrite and Phytogel are also used in some of the commercial labs. The only thing is that Gelrite and Phytogel give very clear media, but it is more expensive than agar. Agar itself is also expensive, but Gelrite and Phytogel are more expensive than the agar. And now overall advantages of tissue culture are high yield, independent of environmental season, and we can obtain the product within short interval of time, or even we can harvest the micropropagated plants within a short interval of time. And then novel compounds can also be obtained. Sometimes the plant is not producing that particular compound we are interested in, but just by manipulating the pathway, not by genetic transformation, just by the manipulation of the media, pH or temperature, we are able to obtain the particular compound. And uniform quality of the products in outside in the nature, if we all the seeds will be different. If you grow the different seeds, it will give rise to different kind of plant. It's if not all, at least 2 or 3 plants out of the 10 will be not the homogeneous in terms of morphological, or especially this affects most in terms of the orchids, the flowers. If you are not able to provide the homogeneous texture or the size of the orchids, then this market there will be no demand of these flowers in the market. And in orchid market, India is the number one to export these orchids to the Holland or the other countries using it by raising through the tissue culture method. And another important point is the year-round availability. It's not like that we have to dependent on the season to obtain the particular compound. In tissue culture, we can obtain the particular compound throughout the year, and it has the potential for low cost production, but it is not very real actually. In the tissue culture, it is one of the aspect in some of the plant species, but actually the cost incurred in tissue culture is very high. So it depends on which kind of plant species we are taking into account. Now showing you the real case studies, the work I have done using this tissue culture method. Coming to the case of chiconin, it is obtained from the lithospermum as already shown you in my previous first 2-3 slides. Arnibia euchroma, it is commonly known as ratanjod. It is a perennial herb and it grows at 3000 to 4200 meter above mean sea level. And its roots are source of red pigment, alkanin, chiconin and their derivatives. They are obtained in the underground parts that are the roots, and it costs $4500 per kg. It is used as a food pigment in coloring clothes, and apart from this, it also possesses antibacterial, antifungal, anti-HIV, and number of other properties, biological properties. This is the slide showing map about the geographical distribution of Arnibia euchroma in India. It is found in the high altitude of Himalayan region, and it's especially in the Himachal Pradesh areas like Lahaul, Spiti, Kullu, Rohtang bypass. This can be seen growing very easily. Now these are some of the commercial products obtained from Arnibia euchroma as one of the ingredient, like ointment, lip moisturizer, balm, burn cream, soap. Now coming to the another plant species, Rheum imodi. It is also known as Indian or Himalayan rhubarb, and it is distributed from Kashmir to Sikkim at an altitude of 2200 to 5200 meter above mean sea level. Its market price is rupees 55 per kilogram. In this case also, anthraquinones, which have number of biological properties, they are obtained from the rhizomes and roots of this plant species. That means the underground part of the plant species, and it also has the antimicrobial, diuretic, anti-cancer, anti-inflammatory properties. This is also the map showing the distribution of rheum at high altitude of Himalayan region, and particularly in the Himachal Pradesh. And these are the common products you might have seen in the market, or some of you might have used also, like for the in Live 55, in Janam Gutti, in Safi. One of the ingredient is Rheum imodi. So now to save the biodiversity, because both of these two plants, they are at the verge of extinction. They are almost going to be rare. So our first priority was to save this plant species. Then another objective was to obtain these components, chiconin derivatives from Arnibia euchroma and anthraquinone pigment in case of Rheum imodi. For this, we initiated the aseptic cultures using young shoot buds, and we tried different media: Murashid Skoog, Linsmere Skoog, and woody plant media, and B5 media at different PGRs. These are the growth regulators for the shoot and root development. We use the number of growth regulators in various combinations. And to set the micropropagation in Arnibia euchroma, the first problem we faced was the contamination. It was not easy to get rid of the contamination because we used the shoot buds also, as well as the buds from the roots also, that were the underground part. And naturally in the soil, the plants, they are close to the number of microbial agents. So to get rid of the contamination was one of the challenge. So we used different kind of antibiotic, fungicides, and then another part was the phenolic exudation. So if there is more phenolic exudation, it will hamper the growth. So in the beginning also, it was not easy to start with the tissue culture of these plant species. And then we studied the shoot multiplication, because we have to standardize the different parameters and each and every stage, starting from the explant selection till the hardening. And then effect of the basal media like Murashid and Skoog, Lensmere, whichever media is best suited for this plant species. We tried different plant media, and then we studied the effect of different PGRs and the status of media, like the liquid media or media with the agar, because agar provide kind of a support, but sometimes it generally is inert, but in very rare cases it also interfere with the nutrients of the plant species, and it interfere with the supply of the nutrients, so the plant growth is not obtained as expected. So we use the media with liquid and with agar also. And in the liquid media, the problem is the hypoxia, because the plants, if they remain in the liquid all the time, they will not grow due to the accumulation of more carbon dioxide. And now here in these pictures, you can see the very nice plants growing through tissue culture. We use the shoot buds in the beginning, and then we obtain these Arnibia euchroma plants in the liquid media and in different combinations of PGRs. Here we tried different PGRs, but the good effect was found in one of the PGR. I'm not going to show the name of the PGR here. And here you can see the difference very clearly. Plants, they are very healthy and green. But because ultimate aim is to transfer them to the outside, so they should be green and stout enough to be survive ultimately to the outside. And here, although the shoot multiplication is little higher than here, but the overall this morphology, you may see that in this case the shoots are little not that green, they are little pale, and they are very thin also, and they are translucent due to the presence of the liquid media due to the hypoxia. So here we selected this media and take it further for the growth. And here you can see growth in the agar gel media. And then after 60 days, we got the flask full of the plant. And outside, can you imagine this flask full, the number of plant just within 60 days? And then after the shoot multiplication, another step is rooting. Here you can see the small root. They are coming out from the shoots. We added the oxygen to obtain the root. Greenhouse for some time covered with the jars because already they have grown in the controlled environmental conditions. Then slowly we took them out to the greenhouse where the light and temperature conditions are partially controlled. And then once they are adapted there, then we take them out to the soil, sand and soil mixture, and then fully to the soil. And here is that was the one aspect of tissue culture, and that was the micro propagation. And now coming to the leaf regeneration. As I mentioned that we can manipulate the plant tissue or cell to any form. Here we tried to obtain the leaves or the full plant from the buds, not only from the shoot or root buds, but from the somatic buds. Like that means from the leaves itself, you can obtain the buds that can give rise to the shoot. So this is very nice discovery that within a single leaf, you can see number of buds and number of plants they're coming out from the single leaf. Outside actually only in bryophyllum species it can give rise to the shoots from a single leaf, but here from a single leaf you can see the number of shoots they are coming out. This is known as the regeneration. And to just check this regeneration, we use the microscopy, and here you can see the cross section of the leaf, and it is showing the vascular bundles and the bud. It shows that there is the direct regeneration. There is no intervening callus form, because if the callus form, it gives rise to somaclonal variation. That means change in the structure, or the how can say in the layman language that the plants obtained will not be the homogeneous one. So in the micropropagation we obtain that is used for the clonal micropropagation, and we obtain homogeneous plants. And if callus phase is there, it will give rise to somaclonal variation, and the plant may not be the same as like the parent plants. But callus phase is must sometimes if we want to reprogram the cell. And now coming to the third application in Arnibia euchroma is the production of this naphthoquinone pigments or the chiconin derivatives. Chiconin derivatives is the particular component. It belongs to the class naphthoquinones, like vinca alkaloids. They belong to the class alkaloids. The chiconin, it belongs to the class of naphthoquinones. And first our aim was to conserve the biodiversity. For that we attempted micropropagation and leaf regeneration. Once the micropropagation is done, we obtain the flask. We transfer them to the outside to the field conditions, and then to the natural habitat or wherever we want to grow them, because in the natural habitat they will grow either in the greenhouse or at their natural conditions where they are already grown at, because they need the particular type temperature and light intensity and the geographical conditions. And now our another aim was, without disturbing the natural or habitat or without disturbing the biodiversity, how to obtain these components from the plant's tissue just using a single leaf, and can also improve the yield also. So here we induce the callus from the leaf. Callus is nothing but it is the undifferentiated mass. If we add 2,4-D in the media, it is normally used for the callus induction, and you see that about 1 centimeter to 2 centimeter we inoculated, that is explant, that is called explant, on the agar gelled media, and after few days there is some protuberance from the leaf and it give rise to callus. Why we need callus? Also, one of the other aim of obtaining callus is because it is very highly growing tissue. Just within a 1 week you will get a this much of material, and we can obtain the compounds from the callus also. But only the disadvantage of using callus is sometimes it give rise to somaclonal variation. It is not that stable. So if our aim is to obtain the fast multiplication, and we can obtain the fast multiplication line also, or the pigment producing line also, we can go in for callus culture. So here are the number of factors that affect the callus growth and pigmentation, because our aim was to obtain the pigment also from the callus, because the factors or the nutrients which we were inoculating or putting in the media, they were not favoring the pigment production, although they were good enough for the callus, because the primary metabolism and secondary metabolism of the plants, they are inversely proportional. For instance, you might have seen that the red color of the leaves in the very stress condition or in the autumn condition, most of the plants they shed their leaves. But if the leaves they remain, they either turn their color to the red or yellow just to protect themselves from the harsh climatic conditions. So at that time their primary metabolism is stopped. That means there is no growth. So they have produced the pigment red color or the yellow color just to protect themselves at that time. That means the secondary metabolism is active and the plant is producing the secondary metabolite. So that means primary metabolite, they are inversely related to the secondary metabolites, or we may say that there is a inverse relationship between primary metabolism and secondary metabolism. So one thing was very clear, that whatever media we are putting here is favoring the growth will definitely not favor the pigment production. So either we have to optimize or choose the media and compromise little with the growth and little with the pigment production, so that there is a one media where can we obtain the optimum production of pigment, as well as we need not to compromise with the growth. But the another way was to devise the two media. First we induce the callus, grow it on the media which is suitable for it, and then take it out on the and put it on the stress media. If we put a give kind of a stress, only then plant will produce the pigments or the specific component. And then in the media we either provide stress in the form of pH, osmoticum, light, temperature, and after that there will be the mass, and only the small lump will be the different color or different shape. And if our interest was to choose the red pigment producing cell line, so within the lump we obtain the little red color slots, and we culture them separately and then try to produce the stable cell line. And with over 2 years of cycle we could able to get the cell line which totally was producing the red pigment. Otherwise, in callus, number of somaclonal variation occurs in every subculture. One we inoculate on the media, and then we need to change the media also. And in every subculturing, we need to keep the pigment producing cell line to the another media. And with this, in the time of 2 years, we were able to obtain red pigment producing cell line, and we culture them and preserve them for the further use. And then from that pigment producing cell line, we obtain the cell culture. Cell culture means those callus lumps we inoculated on the liquid media and put it on the shaker. These are the shakers, and in the flasks here you can say that these are the white in color. That means the callus. For the inoculum or for the biomass, we use the different media, and for obtaining pigment, we culture them in the different media where we obtain the red color. Here you can see the flask full of red color. We presume that this is due to the pigment chiconin. But to see or to test whether it is chiconin or the another component, we did perform some chemical tests. First, we did some just morphological or the qualitative test using KOH, and it gave the blue color there is some chemical reaction, and only the cells which are producing chiconin will give rise to the blue color, other cells will not give. So with this we confirm that it is the chiconin, and when we saw these cells in the microscope, we see the cells, they were all filled with the red pigment. And then to further test this, we went in for NMR HPLC analysis to elucidate its structure, what exactly the structure it is, because there are number of chiconin derivatives. So which kind of particular chiconin derivative it is, we inject these samples for the NMR analysis, and we obtained these two structures which were corresponding to the peak of acetyl chiconin and acetoxy-isoviryl chiconin. And after that, our interest was how to improve the yield of these pigments. For this, we studied the various factors, physicochemical factors. We studied the effect of sucrose. We use different percentage of sucrose in the media. Generally, the preferred sucrose concentration is 3%, but that is favorable to the growth. And what we observed that as we keep on increasing the sucrose in concentration to 12%, the pigment production was increased. So it proved that sucrose, it act as a kind of a stressing agent, because it sometimes also act as a osmoticum, and it also interfere with the plasma membrane and the other biosynthetic pathway of the cell and give rise to the chiconin production, what we observed through this experiment. And another experiment was to check the role of the pH. So we used different pH and found that as the pH increases, the chiconin production increases, but there is a one point at 6.5 where we can, if we keep our cultures at this pH, the growth, we can find the optimum growth as well as the pigment production also within the one flask. And likewise, we studied the effect of light and the temperature also, and what we found that light completely inhibit the production of the pigment. And as we see in the nature also, these pigments, they are present in the roots, which are the underground parts in the soil, and roots, they always remain in the dark. So same way they try to treat themselves as the same way they found in the nature. So what we observed that light conditions totally inhibit the growth of the in the pigment production, although there was no problem with the growth, but pigment we found only when we kept the cultures in the dark conditions. And then we correlated it with the biosynthetic pathway also, and we found that there was a one gene related to the PHP that is related to the dark conditions also, and that gene is responsible for the chiconin biosynthesis production, and that is inducible only in the dark condition. And same way, 25-degree Celsius temperature was found to be suitable for pigment production as well as growth. And now we wanted to see how these cells look like under the microscope. We took out the single cell from this suspension culture and see it under the microscope, and what we see that initially when the cells, they are full of the pigment, they are globular in shape. With the passage of time, the shape, they becoming elongated, and the pigments in the cell, they started moving towards the periphery and then the bulge in the cell appeared, and the pigments, they come out of the cell. But what happened, they just come out of the cell and started adhering to the outer surface of the cell wall. They are not released into the media. That means we need to disrupt the cell to obtain these bioactive components. So that means we need to took our culture or the flask and disrupt all the cells to obtain the media. So we wanted that how to protect the cells and reculture them and just obtain this pigment from the flask, and then again allow these cells to grow. For this, we tried another methodology that I'm going to show you in the next slide. And this is the slide showing the pigments as viewed under the fluorescent microscope and in the confocal microscope, where we can see the pigmented cells under the different layer. And through flow cytometrics analysis, we were able to see the pigmented cells and separated them from the non-pigmented cells, because within the flask, within the cell suspension cultures also, some of the cells, they are pigment producing and other, they were devoid of pigments. So through flow cytometric, we were able to isolate these pigment producing cells also. And then these pigments as obtained from the cells in the flask, we upscale them using the bioreactor, because in the flask we need to change the media manually. Here for the industrial production, we wanted to upscale them. We used the 7.5 liter media in this, and with the 10-liter capacity bioreactor, we tried to upscale this pigment production and all the system oxygen control pH, and all everything was automated. And just within a 12 days, we could obtain this pigment in the flask. So that means our setup method was ready for the commercial purpose also. And now, as I was talking, that this pigments, it remains stuck to the outer surface of the cell. So another challenge was how to regrow these cells, because if these cells, they are producing the pigment, we need to take out the cells and to sacrifice them. But our interest was to how to get these cells for the sustainable production of the pigment, at least for the 3 to 4 cycles. So what we use another paraffin in the media. So because these cells, the pigmented cells, they are lipophilic in nature, the chiconin derivatives. So with the addition of paraffin, they come to the cells, they stay down, and the pigment, it moved towards it's separated from the cells, and it moved towards the upper surface, upper layer. And then upper layer can be easily separated from the other growing cells. So with this, we could save our cultures for 2 to 3 cycles. And these were the pictures of the cells showing in seeing in the microscope. We saw in the microscope by using the paraffin. And what we observed that when this chiconin is, because we tried to put more stressing agents in the media to obtain the pigments, but other way it was dangerous to the cell also, because outside also these components or the bioactive compounds, they are produced in plants in the stress conditions, and that in very low amount. And if the amount of these components are more, these are dangerous to the cells also. That's why they remained in the very membrane bound organelles. And same way in the cultures, they were proving to be the harmful for the cells. So by including the paraffin, we saw that the shape of the cell was intact rather than this, and the cell, they were not they were very good in form, and we were able to reculture them also. Unlike the pigmented cells, they were full of pigments, they were highly globular in shape, and they were not that in healthy. And now coming to the genetic transformation, here our aim was how to reduce, the factors which are reducing the amount of chiconin derivatives, or the other way, how to improve the production of chiconin derivatives by engineering the biosynthetic pathway of this plant species. So for this, we took the callus culture also, and took the Agrobacterium tumefaciens strain using the different segments which are responsible for the growth. We took the same biosynthetic pathway gene from the Arnibia and try to overexpress in the callus, so that we can have a number of copies of this gene which is responsible for the higher production. And then through the PCR, we saw the amplification of the DNA segment, and we use the two different methods, because there are different methods for genetic transformation that I'm not going to go in very detail. One is the using the agrobacterium, and other is the plastic method or the gene gun method. So we did with both the methods, and we took the different bacterial strains to transform it. And so now that was the example with the Arnibia euchroma. And now with I would like to show you the examples of Rhium imodi, another high altitude plant species. Here also, first we try to standardize the micropropagation protocol. We initiated the cultures using the shoot buds, and here you can see the healthy shoots growing in the agar gelled media. This is the agar gelled media, and this is the liquid media. This is also liquid media. The difference between these and this one liquid media, this is the static one, and this is on the shake flask culture. And here you can see the healthy shoots, and then the rooted plantlet, and then transfer of these plantlets to the greenhouse, then to the field conditions. And this is the plant as we obtained after the micropropagation. And in the same way, we wanted to see what are the anthraquinone profile, like whether the plants are producing the same anthraquinone what we are getting in the parent plants. For this, we did HPLC analysis of hardened plants at different stages. That is at after 3 months, after 6 months, and after 9 months. Like once the plants are hardened in the greenhouse, After 3 months, we took the plants and compared with the parent plant. Then after 9 months, and then the 12. And what we found that they produce the same anthraquinone derivatives as found in the natural plant. And here also we made some attempt to make the regeneration. Regeneration protocol is very important, especially if we are interested for the genetic transformation, because our ultimate aim is to transform the full-fledged or regenerate the full-fledged plant from just a single leaf segment. If we change their biosynthetic pathway or manipulate them genetically, then also we need the plant to regenerate after all to obtain the number of copies and then to grow in the nature. And these are the regeneration slides showing regeneration, and also the microscopic pictures to prove that plants, they are regenerated and they are not, there is no intervening callus phase and there is no embryo phase. And then we also compare the callus and cell suspension culture with the HPLC chromatogram, but unfortunately we couldn't obtain the anthraquinone derivatives in the callus or cell suspension cultures. So because these are the undifferentiated mass, so we could relate here that for the production of these pigments, some differentiation is required in the plant species. Sometimes it is not possible to obtain all the components in the callus or cell suspension cultures. No doubt they are fast growing, but for the pigment production, they need some kind of regeneration in some of the plant species. And here we did genetic transformation using hairy root culture. It is obtained by Agrobacterium rhizogenes. Here the beauty of this transformation is that we can obtain only the roots instead of whole plant shoot. First the plant or the seed will give rise to the, or the explant will give rise to the shoot and root. Here directly we can obtain the root using the bacterial strain, because this bacteria has the property to give rise to the roots at the site of infection. Here you can see the number of hairs coming out of the root. That why the name is hairy roots. And these roots are different from the normal roots. As you may see in this flask, they are negatively geotropic behavior they are showing. The natural roots, they always grow towards the gravity, so they are positively geotropic. But these roots are negatively geotropic. They are very fast growing, and if we are interested in some of the compounds which are present in the roots, instead of going in the callus, going for the callus or cell suspension cultures or micropropagation, we can directly obtain the root cultures and can take compound of interest. And this is the all PCR analysis to show that these are the genetically transformed material. And this we did in case of a model plant. This is belong to Solanaceae family, because whatever technique we use in the tissue culture, it is important to test it first in the model plant. Model plant is that grow very fast manner. And now this was all about the plant tissue culture. And coming to the conclusion, we may say that plant tissue culture technology has potential to conserve biodiversity and to obtain the plant-derived specialized metabolite. And it also help us in fulfilling the sustainable development goals, as all suggested these days. All the countries are behind these goals as proposed by the United Nations. But biggest challenge is the to decrease the cost and technical skills. And some of the recalcitrant plant species, especially the monocot plant species, they are very recalcitrant to plant tissue culture, and we need to study their physiology or the genetic makeup, or we need fundamental studies to see the structure of the cells and how we can implement these tissue culture technology successfully in this area. So this was all from my side, and thank you very much. I would like to thank Dr. Meerchand also and the MTTF organization to invite me here and show my research. Thank you very much. And now I may ask if someone has some queries. If you have some queries, you may ask. Thank you.
Thank you so much for this wonderful session, ma'am. It was highly knowledgeable. The way you have dived deep into the plant world, it was so encouraging to all the participants. I would like to ask if any participant have any question. The session is open for questions.
Dear participant, if you have any question, please feel free to ask. You can unmute your mic. You are permitted to open your mic, and then you can put your query. Hardeep Singh, Kuljit Kaur, Mohammed Bakir, Mustafa Grema. Yes, please. The session is open. M Hassan, you can unmute your mic. Anyone, please.
I
Or either you
believe
can put your
ma'am
question in the
has.
chat box also. Okay, no question. Sure, the content which has been presented here as well, recording and the content will be provided. Hasandeep Singh or other participant, we are waiting. If you have any question, please don't hesitate. You can put in the chat box also. Okay, I think, ma'am, there is no more question from the participant.
I guess ma'am has explained the factors very well, so there are no queries anymore. So I would like to thank you again for the knowledgeable session, ma'am. I request all the participants to fill up the feedback form. I'll be sharing the link in the chat box.
Before that, one more. I request to all the participants, please open the webcam for having the group photo, and then after we will share the feedback form and conclude the. I request to all the participants, please open the webcam for having screenshot and group. I request to all the participants, and also, ma'am, I think there is one question from Mayank Tiwari. Does the physiological state or age of the donor plant affect regeneration ability? In the chat box.
Yeah, I can see there is one question if the physiological state or the age of the donor plant affect regeneration ability. Yes, of course, because definitely regeneration it depends also on the choice of the explant and at what physiological state it is. Generally, it is preferred to take the plant at the young stage. If it is already very mature, the cycle is already predetermined, and through tissue culture also, it will not be easy to change its pathway. So it is generally preferable that young explants they are more responsive, so definitely it affects the regeneration ability. And if same way, if the donor plant is affected or its physiological state is not good, then the all the clonal progenies you are going to obtain, it also the quality will not be good. So you have to be very particular in defining or choosing the explant or the initial material at especially its physiological age. Hope I am clear.
Yes, ma'am. Thank you. Dear man, Mayank Tiwari, it is clear?
जी सर.
Any more question? Mustafa Grema, do you have any question? You can unmute. Okay. Okay, ma'am. And now you can conclude the session, Dr. Eparna. No question.
Okay, so thank you so much, Sonia ma'am, for your valuable time and for clearing all the doubts and enlightening us with all the knowledge. Thank you to all the participants for being so patient. And I'll be sharing the link for the feedback form. I request all the participants to fill out that form and certificates will be given accordingly. So, thank you all. So, this is the certificate of appreciation that I would like to present to Dr. Sonia Malik ma'am from the behalf of MTTF. Thank you so much for this webinar in our series.
Thank you, Dr. Panna. Thank you very much, Dr. Merchant.
I have shared the link in the chat box. I request all the participants to fill out that back form.
Okay, may I leave, sir? Okay, I think is.
Thank you, ma'am.
Okay,
Now
thank
you
you
can.
very much for
Yeah, thank you.
inviting me. Okay, thank you, Dr. Panna, also.
Thank you, ma'am. I request all the participants to fill out the feedback form and then they can leave the meeting.